Good morning, everyone. I think we can start, and I'm really happy to be here this morning, and I'm so hopeful that you too. My name is Ilze Elbere. I'm a microbiome researcher from Latvian Biomedical Research and Study Center, and I will be chairing this session. And I'm definitely passionate about precision medicine, and I truly believe that microbiome is an amazing tool for precision medicine. Because as one excellent example, we know that the genetics, what we get, we mostly need to live with it, but microbiome is highly modifiable, highly personalized, so it is a key tool for precision medicine. So that's what we are going to be talking today. And I hope that after these interesting and amazing talks, you will also be more excited about microbiome research and more passionate that microbiome can be useful in precision medicine. And I will also have a co-chair, John Penders from Maastricht University Medical Center. He will also be helping in introducing the speakers and the questions preparing. And as in other sessions, so firstly we will have the presentations. If you have some questions, write them down or try to remember them at the end when we will have the Q&A and discussion for all the speakers involved. And as the first one, I would like to invite Mathieu Almeida from Ilze. So please, the floor is yours.
Good morning, everybody. First of all, thank you very much from the PMNET Forum for letting me introducing this work that I'm doing in France and colleagues around the world. Today I'm going to talk about the way to explore the microbiota and how we can use the discoveries in fecal sample analysis to help improve the screening of colorectal cancer. So I'm going to talk about microorganisms that you can find in high quantity in all ecosystems, on the ground, on the food, on yourself, and inside you. Depending on the ecosystem you're looking at, the composition is a little bit different. From the saliva, which contain a lot of aerotolerant microorganisms, to the intestinal microbiota, which are more anaerobic in healthy conditions. On the right of this slide, yes, that's cool. I included the microorganisms that you can find in the fecal samples when you analyze them. You have many type of microorganisms, mostly bacteria, really dominant in the fecal samples, like Akkermansia mucinifila. You also have the phages associated that are attacking these microorganisms, or human viruses as well that you can detect. Archaea, it's not in everybody, but you can also detect archaea microorganisms involved in the processing of methane. And you also have karyotic microorganisms like fungi or protozoa. And for sure, in the fecal sample, you also detect human cells. And sometimes in this biosis, you have a lot of human cells detected. It's actually kind of the fit test for colorectal cancer. When you have blood, you also have human DNA with it. And this is the kind of signal we can also detect with the technique I will show you. In the past years, we saw that we have a constant decrease of infectious disease coming from tuberculosis, of hepatitis, but in parallel, you have a bloom of chronic disease like Crohn disease, type 1 or type 2 diabetes, asthma. And the question is it related to our lifestyle, antibiotic usage? Is it impacting our gut microbiota? And is the gut microbiota preventing or reducing the risk of developing some of these diseases? That's the main question for our topic. To explore the fecal samples, you have a lot of different techniques. Commonly, you can extract DNA and focus on the DNA. That will answer what are the type of microorganisms that you can find, what's their abundance, because sometimes the abundance is important for triggering an inflammation, for instance. low abundance can sometimes don't have any effect. You can also have an inference of what they can potentially do based on the genes that you can find. But having the genes do not say that they are expressed. You just see that they are there. You don't know if they are expressed. And that's why you have other techniques like metatranscriptomics or metaproteomics or metabolomics that give you an idea of what is secreted, what type of proteins that you have that have with the protein has the function to trigger a specific pathway of inflammation, for instance. But today, I will more specifically focus on DNA exploration. And for DNA exploration, you have two types of exploration. You have amplified specific regions of the DNA, like the 16S, or the ITS or 18S. This is really interesting to amplify because it's cheap usually and easy to analyze, but the problem when you do that is that you only analyze a specific type of microorganism, like prokaryota, for instance, or fungi. You don't have a full picture at the same time. So then we have metagenomic sequencing so randomly sequencing everything you can find. This is really interesting, but it's more expensive in comparison, but it gives you a lot of information. With the money you put, you have a better resolution of the microorganisms you find. You can go down to the strain level, and you have a lower relative abundance bias because you don't have to amplify a specific region. You take everything you see, and you have a kind of a relative abundance that is respected. But it's more computer intensive, so you need resources for that. And it's a little more expensive, but it really depends on the number of samples you analyze. So today I'm gonna talk more specifically about metagenomic sequencing. With metagenomic sequencing implies that you have to master different steps to have the best analysis at the end. It means that when we do analysis, if the collection step is not well done, we miss the important signal. So the way you collect is the key part of everything. Then how you do DNA extraction is also a very important part, because when you do DNA extraction, you have to make something that is good for not only prokaryota, but also for eukaryota, for instance, fungi, as much as possible. And at the end, using sequences, it's the depth of sequencing you will reach. It's like a picture you take with your photo, right? If your photo is of high resolution, then you can zoom in. And by zooming in, you see things that you cannot see when you look by distance. So exactly the same. Sequencing more, increase the resolution, and you see low abundant species that could be related to the disease you're analyzing. And then you have reference database of genomes and genes to annotate the molecules that you sequence at the end. A very traditional way to do analysis of microbiota or meta-analysis is to centralize the data coming from different regions around the world. So imagine you have publicly available data, or you also work with colleagues in looking at colorectal cancer. So what you do is you have agreements with your colleagues. With them, you also have the ethics from the patients to centralize the data somewhere. So sometimes the somewhere could be outside of your country. It means that in that case, you trust your colleague that they will protect this sensitive data for you when they analyze, right? So this is really a classical way to do things, because sometimes you don't have the expertise in your team to really analyze the best way possible the samples. So centralizing the data, you trust someone, usually someone that is got the knowledge of analyzing things, and at the end, you and your colleague is in charge of securing the data. This could be a high risk for a privacy breach or secure data breach, for instance, because if the center is not really protected against hacking, then all the data then could be a breach, and they can take everything directly. And also because of that, most of the time when you work with colleagues, you don't necessarily want to provide all the information you collected, because this is really precious, right? You can have blood samples, you can have fecal, saliva, all these things, but for the study, for instance, you just want to provide a part of the information because you want to do other things with these samples for other studies. So you only share a part of the information, but that's sometimes a shame, because having blood information, for instance, can provide information, new information that improve the result that we have at the end. So this is an example of such traditional meta-analysis. So there is a team expert in Trento in Italy that gathered all the information from different publicly available data. So these are the cohorts that were published that they used. So they merged them, they standardized everything for the analysis. And as you can see on the groups, you have a lot of different type of phenotypes. Control, so healthy individuals, asymptomatic, some with adenomas and some with colorectal cancer. Usually it's about 50-50 in male-female, but in colorectal cancer, there is a little more men than women affected by the disease. So there is an imbalance between them. And most of the time, as it's complicated to get colorectal cancer, it's a small data set. And at the end, you also have missing information, because sometimes it was not collected prior to the study. So even with these difficulties, you can still do meta-analysis. And that's what this expert-centered team made. They used this sample and they identified the microorganisms that are associated with sick or healthy individuals. In this graph on the top, with the red colors, you have the fecal microorganisms that are most of the time used by machine learning method to stratify the individuals if they have colorectal cancer or if they are healthy. So you have a software that is based on the composition, deciding that this is a healthy profile or a colorectal cancer profile. And the number you have in the cells is the number of the ranking of these species in the models. If it's close to 1, it means it's a key microorganism in identifying or stratifying the individual if it's colorectal cancer or healthy individuals. And as you can see, Fusobacterium nucleatum is one of the top microorganisms in these models. Fusobacterium nucleatum is already today known as a potential cause of development of tumors in the intestinal tract. So it makes sense that this is actually one of the biomarkers for stratifying individuals at risk or not. So what they did after that is they used these species to classify the individual if they are colorectal cancer or not. This is an AUC visualizing. When it's close to 1, it means that you are perfectly separating individuals that are colorectal cancer or healthy. So what you want is one classification for each of your cohort. Each line is one of these cohorts by colors. And each numbers on the bottom is the number of biomarkers you use to classify. Only 2 species, or 4, or 16, or all of them. And as you can see, when you use a lot of biomarkers, you improve the power of your classification. It's not dramatic, but it's still improving close to 0.8 in accuracy, which is really nice. But sometimes, some cohorts, you do not have this power of classification. So one of them is still in the bottom, even if you use more species. And after looking at this cohort, more specifically, we saw that this cohort, the collection process and the DNA extraction process was not the best, using the best standards today. So it means that when you collect the samples, if you collect it with the best standards, then you can get the best out of it for the classification. If not, you will have troubles to classify the individuals, unfortunately. So now, sometimes it's not possible to centralize the data because you don't want to, you cannot, by your ethics, send the samples outside and the metadata outside of your country. So this is a classical now, more classical with our GDPR rules. These are actually protecting the patients to anonymize them as much as possible. So that's one reason that sometimes it's not possible to centralize. And actually there are techniques to make this kind of analysis. It's called federated analysis. It means that instead of providing your samples and your data, you receive the algorithm in your lab to process the samples. And at the end, what you share is not the data, is just the variables, the species and their abundance that are really important to classify. For instance, you say, in my cohort, it's Fusobacterium nucleatum that is one of the best predictor. You share that information with another team, and they share with you their biomarkers, and now you classify with their information. And at the end, you can, by this way, collaborate in providing information of what is really important in your data. And what is cool with that is because you don't share these data, you can input a lot of different things without the risk of data privacy breach. Because the data is located in your lab and is just the result of the algorithm that is shared. It could be age, it could be a blood inflammation biomarkers at the end. And so we have a project of federated analysis with different colleagues around the world, including a team in Ireland for that. It's called Microbiome. And we've made such federated analysis with different samples around the world. And so in this graph, you have a comparison between the classical, traditional way of doing classification between colorectal cancer or healthy on the very left. This is the centralized techniques. It's actually one of the best methods to stratify, but sometimes you cannot do that. So you want to do federated analysis instead. And what you can see is with federated analysis, when you work with one hospital only, or two hospitals, or all the hospitals, together you improve the classification. So that's a way to really improve the analysis of colorectal cancer screening without sharing the data, but working together. And though my presentation is done, I just wanted to mention that in France we have such large-scale federated analysis with my colleague Robert Benamouzik, aiming for 2,500 individuals. And at the end, so gut microbiota is really powerful to identify the person at risk of developing colorectal cancer. But sometimes you cannot centralize the data with colleagues. So federated could be a way for that. So this is the team that is behind all this analysis. And thank you very much for your attention.
I'm happy to announce is John Penders. He's a researcher at Maastricht University Medical Center, a professor in intestinal microbiology and metagenomic epidemiology. And one of his main aims is to use microbiome to distinguish healthy cases from disease. And I know that a passion of John is antimicrobial resistance, but today he will be asking the question, is gut microbiome the missing link in personalizing the colorectal cancer treatment?
Thank you. Thank you for the introduction and also for the invitation to present here today on this very interesting conference. And like said, I will be focusing today on colorectal cancer. Just to show, this is our institute over here. So we are located in the University Medical Center, so we have a direct link with clinical departments, and we also try to implement microbiome research and see how it can help patients. Now, we didn't agree on this before, but I just wanted to show how microbiome can be used in cancer-based diagnostics, and there are different aspects related to that. And this is exactly the example that was already mentioned by Marcus just before, using the microbiome to detect and diagnose cancer. And we just heard how difficult that can be, especially if you use samples that are low in biomass and often very difficult to detect the microbiome. So this is not what I would like to talk about. I would like to talk about the use of the microbiome in predicting treatment response and treatment side effects, a field so-called, also referred to as pharmacomicrobiomics. And just to unravel the terminology pharmacomicrobiomics, as the word already says, it includes pharmacology, it includes the microbiome, and it includes omics. So using omics-based approaches like metagenomics or other omics-based approaches to profile the microbiome and see how that is related to pharmacokinetics, but also pharmacodynamics. And if we then look at the different actors and the different levels at which the microbiome can be involved in interactions with drugs, we on one hand have the impact of the drugs on the microbiome, so it can inhibit certain microbes, it can promote the growth of certain microbes, but it can also result in different metabolism in these microbes, or even result in over or underexpression of virulence factors. And on the other hand, we have the impact of the microbes on the drugs. And that can be that microbes can accumulate drugs so that they are not available for the host, so xenobiotic accumulation, but also metabolize certain drugs. And especially this last part is something that I would like to exemplify in the next 15 minutes. So we questioned whether the gut microbiome is the missing piece in the puzzle to optimize cancer chemotherapy. And we know that if we look at what has already been done in patient profiling prior to treatment, we often look at tumor characteristics, we look at the patient's genotype in certain treatments, we look at the physical condition, but we don't look at the gut microbiome. And is this something that we can add to optimize treatment responses, but also especially to prevent certain side effects? Now, I would like to give two examples today of two drugs that are being used to treat colorectal cancer patients that have metastasized colorectal cancer. So these are treatments not given as a curative treatment, but to extend their life expectancy. And you can imagine that in these groups of patients, it's very important to make sure that they have sufficient quality of life. So you don't want them to have severe side effects of these drugs. So the drugs that I will present today is capecitabine, and the other one is irinotecan. And here we see capecitabine. So it's given as an oral drug in an inactive form. It's being converted into different steps in the liver and later in the tumor cells. And the active drug is 5-FU, and thereafter it's inactivated again in the liver and secreted. And especially at present, we genotype patients for the DPYD variants because certain patients cannot inactivate the drug, and of course they will have severe side effects. Now, there have been many studies that have already looked into the impact of drugs on different gut microbes, and especially the group of Lisa Meyer in Germany has done excellent work on that. So what she did is she screened a panel of over 1,200 compounds against 40 different bacterial isolates. And what she showed is that if you look at the effects, almost all, or at least the vast majority of antimicrobial drugs also impact commensal microbes, so almost 80%, and you see that in the green bar in this slide. But she also showed that 24% of the human-targeted drugs also have an impact on at least one of these 40 commensal microbes. So a lot of human-targeted drugs also affect the microbiome. And if we then look at 5-FU, it was shown that 26 out of the 14 strains were inhibited by this drug. So clearly showing that this drug has an impact on the microbiome. But other studies showed also the opposite. So this is a study in which they incubated E. coli or Salmonella enterica with 5-FU, and what you can see in the graph over here is that if you add E. coli to this medium with 5-FU, you see in blue the conversion into the inactive metabolite, and we see the same for Salmonella. So E. coli is able to inactivate this drug in the same way as is being done in the liver. And they also identified the gene responsible for that, and this is the pre-TA gene in this E. coli strains. They did a lot of follow-up experiments with knock-out strains of this gene, and they could indeed show that if you knock out this specific gene, then E. coli cannot inactivate this drug again. So clearly showing that bacteria can also play a role in inactivation of this drug. They did a follow-up experiment, and what they showed here is they did a mouse model with mice with tumors, and they treated them either with capecitabine or they didn't give them a treatment, and then they added an E. coli, a wild-type E. coli, a mutant E. coli, or a wild-type E. coli. And what they could find is that only if you give capecitabine with the knockout E. coli, then you see a decrease or at least a stabilization of tumor volume. But if you give capecitabine together with the wild-type E. coli, you don't see a treatment effect anymore. So E. coli can completely abolish the effect of the treatment in this mouse model. Now this is nice, of course, but this is all in vitro and animal studies, and there's still quite limited evidence of what is happening in the human context. And this is something that we have now started to do in collaboration with our clinical department and with our postdoc Janine Simos. And what she did is she initiated a cohort study of 44 patients that were treated with capecitabine, and she collected stool samples at baseline and then again during the 3rd cycle of chemotherapy and prior to the 4th cycle of chemotherapy. And one of the first things that she noticed is that if you look at the short-chain fatty acid levels and the branched-chain fatty acid levels in the fecal microbiota of these patients, that patients with lower levels of the branched-chain fatty acid isobutyrate had the best outcome. So they had a partial response, whereas progressive disease and stable disease had higher level of isobutyrate. Now this shows that the microbiome might be able to predict treatment response. This is still very subtle findings, so not very robust. But we wanted to move further and especially focus on treatment of toxicity, because we think there is definitely the most to gain. And we did this in collaboration with a group of Peter Turnbaugh in San Francisco. We did metagenomics on this cohort and had now a number of 56 colorectal cancer patients. Same design. And first, of course, we looked at this pre-TA gene because it has already been shown to be impacting treatment response in mice. And we could show that if you look at the PTA gene in the microbiome of patients, you see an increase during the course of treatment. So at a 3rd time point, the levels were significantly higher than as compared to baseline. But more importantly, you show that patients at baseline with the highest level of this pre-TA had less dose adjustments. So this is a little bit opposite to what was shown in the mice model, that if you have this pre-TA gene at a high abundance, dose adjustments were less necessary than for the other patients with low levels of pre-TA. And we then also looked at toxicity and side effects, but we couldn't find an effect of pre-TA abundance on peripheral sensory neuropathy and also not on the hand-and-foot syndrome, common side effects in these patient groups. But then we went further and looked in general to the pathways that were encoded by the microbiome, and we could see that there were certain pathways here in the volcano plot depicted in blue that were enriched in this patient population, and 7 out of the 10 most enriched pathways were related to minocanol, so vitamin K, and we also showed that up to 70% of the patients had increased relative abundance over the course of treatment in these minocanol-related pathways. And also, these pathways were mainly encoded by E. coli, as you can see in the second graph, and you can also see that the enrichment in these pathways during the course of treatment was mainly due to an increase of E. coli in this case. So we then looked into to what extent do these different pathways also predict toxicity and side effects. And first we looked at the overall microbiome and all the pathways encoded in the microbiome, and we could indeed show that if you look at this figure, that the variation in microbiome pathway abundance was able to predict significantly peripheral sensory neuropathy, alopecia, and oral mucositis. And then we moved on to the minocanol-related pathways again, and saw that especially for peripheral sensory neuropathy, it was shown that patients with higher abundance of this menacanol-related pathway had less of these side effects. Of course, these are only genes, so we also looked at the metabolite, and we could indeed see if we quantify this metabolite in the feces, this was also higher in the patients that didn't experience side effects later on. And then we wanted to mechanistically prove this effect, and we could indeed see if you give mice capecitabine and look into effects of peripheral sensory neuropathy done by the hot plate test. So mice are put on a plate at baseline and again at the end of treatment, and the speed in which they lift their paws is an indication of this sensory neuropathy. And if you give capecitabine together with menacanol, you see that you can basically revert the sensory neuropathy that is being induced if you only give capecitabine. So here we show that indeed giving this metabolite can, at least in the mouse model, revert these side effects. And hopefully this is also something that we can induce in humans. So now we're going to focus on a follow-up study in which we try to manipulate the microbiome of this patient population using prebiotics in the hope that we can also affect their side effects. So this was the first example. The second example is irinotecan metabolism. So this is a study where we don't have that much data on yet, but this is definitely an example of a drug where I think translation to the clinic to reduce side effects is most strong so far, because we know that irinotecan given systemically is in the end converted in the liver into SN38G, so it's conjugated to glucuronic acid to inactivate it, and then it's secreted through the bile into the lumen. But the problem there is that this inactive compound in the lumen can be reactivated again by the bacteria there, by so-called beta-glucuronidases, enzymes encoded by bacteria. And this reactivation in the lumen by bacteria can result in gastrointestinal toxicity. And these are very sick patients, and you can imagine if they get severe gastrointestinal toxicity, that this can really impair their quality of life. We see that patients need hospitalization, they are dehydrated, often develop mucositis, colitis, they have an increased infection risk, and definitely a decreased quality of life. And often this leads to dose reductions and also often treatment discontinuation. So this is definitely something that we don't want in these patients. And we know that the cause is in the microbiome in this case. There have been a lot of in vitro studies, and they already showed that these beta-glucuronidases, this is a whole family of enzymes, and a certain group within that family, the loop 1 beta-glucuronidases, are particularly involved in deconjugating this SN38G. And also in mice studies, they showed that if you give mice this treatment, so irinotecan, combined with a beta-glucuronidase inhibitor, you see that the treatment efficacy is not affected, so the treatment efficacy is still there, but you see that if you add this beta-glucuronidase inhibitor, you can significantly reduce diarrhea and gastrointestinal toxicity in these mice. So also here we want to translate this to the patient population. We have again a patient population of metastasized colorectal cancer patients with longitudinal fecal sample collection. And what we're currently doing is setting up an enzyme assay to measure the conversion of SN38G in fecal samples of these patients, as well as looking into their metagenome and build a beta-glucuronidase reference database to which we can map the metagenomes of patients in order to predict to what extent they have these enzymes and to what extent they are prone to future gastrointestinal toxicity symptoms. So with that, I would like to conclude, and I hope I give you a little bit of insight in how the microbiome not only can be used in treatment or in diagnosis, but maybe also in treatment response, and especially also in predicting treatment toxicity in order to better personalize treatment to this group of patients. Finally, of course, I would like to thank all the people involved. As you can see, many people are involved in these kind of studies, not only researchers, but especially also a lot of clinicians and a lot of hospitals to recruit all these patients. So I would like to thank them for that, and I would like to thank you for the attention. Thank you. Okay, and now we move on, and I have the pleasure to welcome Dr. Monta Briviba, if I pronounce it correctly.
It's fine, thanks.
And she's the head of the Genome Center at the Latvian Biomedical Research and Study Center, and she will be talking today on the from resource to discoveries, gut and oral microbiome in pediatric cancer.
And before Monta moves on with her presentation, I would also like to emphasize and share that just a couple of weeks ago, Monta received the Baltic Awards for Women in Science, also on the topic of precision medicine in oncology.
Thank you. Good morning, everyone. It's my pleasure to introduce you with a relatively recently initiated project, which is a collaboration project between Latvian Biomedical Research and Study Center and Children's University Hospital, focused on microbiome analysis in pediatric cancer patients. And I will start with a brief introduction back into history. So our collaboration in the field actually started back in 2019 when the Latvian Pediatric Cancer Initiative was launched, and the main aim of this initiative was to start biobank, biospecimens in the National Biobank, and also do the next generation sequencing, meaning whole genome, whole transcriptome sequencing for these patients. And it has led to more than 500 patients recruited in the biobank today, and around 200 of them fully sequenced. And of course, it has also led to many really productive collaborations. And on top of that, in 2024, basically thanks to a few really enthusiastic doctors from Children's Hospital, we also agreed that we will start to collect microbiome samples from the same patients. And we started with stool samples and then also decided to add oral microbiome samples. And today we have these microbiome samples collected from more than 80 patients, and we have done the gut microbiome analysis for 34 of them in at least one of the samples collected. And in addition to that, this year, we also started to recruit their healthy siblings in order to use them as healthy controls in the near future, since they are sharing the same environment. Okay, and the microbiome itself is still a hot topic in different fields, and childhood cancers is not an exception. You can see many nice papers out there linking both the gut and oral microbiome with diagnosis subgroups, with cancer therapy, with specific pathogen infections, and also with the microenvironment of the tumor itself. But the main aim of our study is a bit broader. It is to characterize the gut and oral microbiome in children with cancer and identify microbiome-based markers that would predict disease course, treatment response, and complications. And to achieve this goal, we developed a few objectives. First of all, we want to build a biobank and database, and this is the state where we are currently at. Then we want to profile gut and oral microbiome composition, which we started to do relatively recently. Then we want to identify and validate these biomarkers that could be linked to prognosis, therapy response, and potentially also complications. And finally, we want to pinpoint disease-specific microbiome markers by comparing the microbiome profiles between cases and controls that in the future will be these healthy siblings. So I believe that my duty is to promote my workplace, so I'll just, for those who are not aware of it, these samples are being collected in the National Biobank genome database of Latvian population. that is maintaining different types of biospecimens from more than 43,000 individuals at the moment, and we are also serving as the national node of the BBMRI ERIC infrastructure. And in addition to that, all of the molecular analysis, including and mainly next-generation sequencing applications, are being done in our core facility, Genome Center, Laboratory of Molecular Genetics. So now we'll go back to the study design. We decided to do a longitudinal study, meaning that we are doing multiple sampling times during the therapy from each patient. The first sample is being collected before the therapy, and all the rest samples are being collected during or after the cancer therapy. And our colleagues from Children's Hospital are really working hard to collect clinical lifestyle data and also food frequency questionnaires, and this is still in process also for the cases that are already recruited in the biobank. And as I said, we have also already recruited more than 80 patients now that have donated almost 700 stool aliquots and more than 300 buccal swabs. And you can see distribution here, distribution of these samples across different visits, and you can see that's highly heterogeneous, but it was kind of expected due to this dynamic course of the disease. So about the methodology, for gut microbiome analysis, we are doing shotgun metagenomics using the DNB-seq G400 sequencer, and for oral microbiome, due to the presence of the host DNA, we are doing more targeted approach, which is 16S ribosomal RNA gene hypervariable region sequencing, and this is being done on the MySeq platform. So the data analysis is done using open access tools and our in-house pipelines, and we are doing the data analysis using our own computational resources as well as using the Riga Technical University HPC Center. And before I jump into the results, I just wanted to emphasize that the population of childhood cancer patients is highly heterogeneous, and this is due to different diagnoses and different subtypes within each of the diagnoses. And this means that we need a lot of samples to do any kind of analysis here. In addition to that, we decided to do a longitudinal study, which kind of means that each patient serves as a control to him or herself, which helps us to improve statistical power. But from the other hand, it means that we need time to collect the full sample set from each patient. And altogether, it means that we need a lot of time to build a research to do this microbiome analysis. And now you'll see really preliminary results. These results cannot be used to make like confident assumptions. Nevertheless, we can use those results just to predict what we would expect, what we could expect from the upcoming analysis. So these were the gut microbiome profiles from 34 childhood cancer patients that were followed up longitudinally, plus 22 healthy controls. And these controls are not the healthy siblings that I mentioned before, because we started this arm relatively recently. These healthy controls are coming from a different project. These, despite being kids, they are a bit older than our patients, which must be taken into account when interpreting the results. So here you can see a representation of different diagnosis groups among the sequenced cases, and also representation of samples across different, across the time points and the visits during the therapy. And you can see, again, a huge heterogeneity there, the same as it was in our previous slides. So initially, we wanted to identify the factors that are influencing the gut microbiome composition in these patients. And we are aware that at that point we didn't have all of the necessary clinical and lifestyle data, but we did our best. And what we saw was that actually patient itself is explaining around 42% of the variability observed in the gut microbiome, which is kind of expected. And it was followed by a diagnosis explaining around 4% of variability and age explaining around 3% of the variability. I was really happy to see that already in this preliminary data with relatively small sample size, we can see that the diagnosis is a significant factor itself at the baseline. So then we focused on the baseline samples, meaning the samples obtained before the therapy. And you can see in the panel A where these data are not supervised, these are multidimensional, this is a multidimensional scaling plot. You can see that controls are kind of clustering together already while the disease samples coming from cases are kind of scattered around. So then we, in the panel B, we added some covariates, including the information about different diagnosis groups, and you can see clustering there even more clearer. So then we wanted to see what is happening in the diagnosis samples solely. So in order to improve the resolution, we kind of got rid of the control samples in panel C, and despite the limited sample size, you can see that microbiome profiles coming from the same diagnosis group are kind of clustering together. Unfortunately, I have no pointer, so I cannot really show you, but I'll do my best. You can see like the, the blue triangles here are coming from central nervous system tumor patients, while there are purple circles, those are coming from solid tumors, for instance. So then we wanted to see what is happening with this intersample diversity longitudinally. So we focused on time points 1 to 4, which is around 1 month in time. And in panel D, you can see Bray-Curtis distances in the gut microbiome profiles in each of the visits compared to the baseline, meaning compared to the samples obtained before the therapy. And you can see that in those box plots, despite this huge heterogeneity, you can see that these distances are increasing in time, meaning that this gut microbiome composition is kind of diverging during the therapy, at least within this month period. So then we wanted to see what is happening with alpha diversity. So here you can see Shannon index. In panel A, you can see clearly that the Shannon index in the gut microbiome profiles coming from cases is smaller than compared to controls. And when you stratify the cases in different diagnosis groups, you can see, first of all, that the sample size is a huge limitation for us in panel B, but secondly, you can also see that it kind of differs between different diagnosis groups. So then we wanted to see what is happening with alpha diversity longitudinally. Again, we focused on the same 4 visits within 1 month period, and we were expecting to see like a drop or decrease in this alpha diversity, but I guess that our data set is not really reflecting that. So we wanted to clarify what are the specific species that are determining this gut microbiome variability. And we compared cases against controls at the baseline, meaning before the therapy. We did this differential abundance analysis, and we observed 14 differentially abundant species there. You can see in the heat plot that basically all of them had a lower abundance in cases compared to controls. And here's just one of examples, this Agathobacter rectalis. This is considered a beneficial bacteria, it's butyrate-producing bacteria associated with Mediterranean diet, and it's negatively associated actually with colorectal cancer. You can see that it has lower abundance in hematologic malignancies compared to controls. And then another thing that caught my attention was this kind of spike in 2 central nervous systems cases of Clostridium inoculum. And I kind of noticed this bacteria because it's an opportunistic pathogen, and you can see that the abundance of this pathogen is increased only in 2 cases, and this opportunistic pathogen has been previously linked with infections of Clostridium difficile, which is a well-known pathogen also in pediatric cancer cases. So we wanted to see what is happening longitudinally. Again, we compared the same 4 time points, but in order to make the patient group more homogeneous. We focused only on hematologic malignancies. We did the differential abundance analysis comparing each of the time points with the baseline, and we identified 8 differentially abundant species there. And again, just a few examples. Bacteroides uniformis in panel A, you can see that after the therapy, there is a drop in the abundance, which kind of gradually increases. And completely opposite situation in panel B, the same Clostridium inoculum that I mentioned before increases right after the therapy and then again gradually decreases. And now I will shift to oral microbiome. So as I mentioned before, we started to collect the oral microbiome samples much later than we did with the stool samples. So there are less patients available, and unfortunately we do not have controls yet to compare with. Nevertheless, you'll see just a small insight in the results coming from 22 childhood cancer patients that were followed up longitudinally. Again, just a general representation of the diagnosis groups across these samples and a representation of the samples across different time points during the therapy. So what we saw here, so in oral microbiome analysis, we focused on specific diagnosis groups and followed them longitudinally only since we didn't have any controls. And what we saw in hematologic group is that there were no really clear patterns in alpha diversity in panel C during the therapy. Nevertheless, we saw in panel A that after the first visit, after the therapy, there was an increase of Streptococcus genus, which gradually decreased. And the Streptococcus appeared also in a differential abundance analysis, and also in panel B, you can see that Streptococcus is a significant factor when we're talking about intersample diversity. Similarly, we did the same analysis in solid cancers, and we had fewer solid cancer cases than we had hematologic malignancy cases. So probably that's the reason we're not getting really statistically significant results, but you can see like a tendency here in plot C that right after the therapy we see a decrease of number of features and Shannon index, which we didn't observe in hematologic malignancy group. And if we're looking at the taxonomic composition, there's the same situation that right after the therapy in panel A, there was increase in Streptococcus genus, which gradually decreases. So since we observed these differences between hematologic malignancies and solid cancers, we kind of decided to compare the subgroups, and we compared the oral microbial composition between hematologic malignancy patients and non-hematologic malignancy patients. So we put all of the rest of the diagnosis in a single group just because we had a few samples there. And indeed what we saw was a reduced number of features in the panel C, a reduced Shannon index, and interestingly increased evenness in hematologic malignancy patients compared to non-hematologic malignancy patients already at the baseline, so before the therapy, which altogether means that the diagnosis itself is a really significant factor in both in gut microbiome and oral microbiome, and I'm really happy to, can't wait to see what is coming out from the following analysis. And here are some just list of the future perspectives. As I said, now we are focusing on building the microbiome and clinical data research, but we are doing microbial pathway analysis. Just that I didn't manage to put the results in this presentation. My colleagues are working on the pipeline development for antimicrobial resistance analysis, which is crucial in these patients. And we are also collecting samples in appropriate preservation solutions to be able to do stool metabolomics afterwards, but specific targets, metabolite targets will be selected according to the results of the microbiome study. And this is our team. Thank you very much for your attention, and many thanks to patients and their parents as well.
Thank you, Monta. And the final speaker will be Valeria Movčana. She is a researcher at Cellbox Lab, a Latvian startup company focusing on organ-on-chip development. And she will tell us more about gut on a chip.
Yes. Hello, everyone. My name is Valeria, and I'm a researcher at Cellbox Labs, where we are developing industrial organ-on-chip technology. And I'm really glad to be here today with all of you and introduce you about our gut-on-chip platform and how it can be used for anaerobic microbiota host communication studies. Yes, so what are organs on chip? Organs on chip are miniature organ replicas or in vitro test models that can be used for testing various biological applications, like drug delivery, disease modeling, microbiota research, and more. And these devices mimic human organ functions, providing more accurate experimental models compared to traditional methods. Okay, yeah. Yeah, for example, current in vitro model systems like well plates or inserts fails to replicate complex gut physiology, lacking the dynamic environment necessary to mimic in vivo conditions, such as fluid flow, mechanical forces, and complex cell-to-cell interactions. And as well, these models are not suitable for microbiota research. Therefore, organs on chip enables the development of dynamic 3D cell culture models. And as we can see here, organs on chips consists of vertically stacked channels, and they are separated by the porous membrane, which allows nutrient flow, waste removal, and cell communication. Yeah, so about this process, we can seed cells into both of the chip channels to create vascularized models. For example, with the micropipette, we can add epithelial cell suspension in the top channel and endothelial cell suspension in the bottom channel. Afterwards, chip needs an incubation period in the CO2 incubator for the cells to attach to the membrane. And when it is confirmed with bright field microscopy, then we can perfuse laminar cell media through flow through microfluidic channels. Why this laminar flow is so important? It's because it impacts cell morphology and cytoskeleton organization. For example, here is the endothelial cells in the static conditions versus endothelial cells after laminar flow has been applied. Also, with intestinal cells, laminar flow allows to develop 3D pseudocrypt and microvilli structures. If we are talking about advantages of these systems, I want to say that there is always like a trade-off between reproducibility and physiological relevance. Because simpler systems like 2D cell cultures, they are easier to use, but they are less realistic, while complex models like animals are more realistic but harder to standardize. Therefore, organs on chip aims to balance both. Because there is no fluctuation in biochemical gradient due to media change, like in other in vitro models, and also it is possible to mimic physiological conditions, like shear stress, oxygen gradient, intestinal peristaltic vasculature, and other. And of course, there is opportunity to create multi-organ systems where we can create different organs in controlled environment. But handling of these organ-on-chip systems can be challenging because usually these setups consist of many components: syringe pumps, syringes, chip connections, and tubings for the liquid delivery to the cells. And that means to messy and error-prone configurations, inconsistent flow delivery, which also affects flow precision and therefore reproducibility of the result. And of course, these complex setups can increase contamination risks and require specialized lab, intensive user training, and these systems are not easily adapted into multidisciplinary labs. So we in Cellbox Labs developed the all-in-one system. This system consists of three main components, which reduces hands-on time, error rate, and is in general more user-friendly to get more reproducible results. So we have our chip where we seed and culture the cells. Then when the cells are attached and it is confirmed, the chip is inserted into the manifold, and manifold reservoirs are filled with the cell culture media. And the manifold is the component which ensures this flow and has all of the sensing elements, like flow sensors. And then a manifold is inserted into the instrument where it connects to the pressure system for flow delivery, And afterwards, this instrument can be controlled with software, and there is also integrated real-time bright field microscopy, and you can control microenvironmental parameters like change flow rates, temperatures. And now I will introduce you into our developed model, which is gut-on-chip. We developed this model with KCO2 and HD29 MTX epithelial cells in the top channel, and in the bottom channel we cultivated HUVEC endothelial cells. And to confirm successful model development, we examine marker expression, and in the picture A, we can see in red Willen expression, which is the brush border marker of the intestine, and also in green there is MUC5AC expression, which confirms goblet cells in this model. If we are looking at the picture B, where is the bottom channel, there is fully confluent endothelial cell layer expressing CD31 in green, which confirms the endothelial cell identity. Here are also confocal microscopy videos where we can see all of the epithelial marker expression, and I would say a really dense epithelial tissue layer. To confirm that we have mucus production in our chip, which is really important if we want to research microbiota, we applied Alcian blue and pus staining to identify neutral and acidic mucins. And we established two co-culture ratios. We used KCO2 enterocyte-like cells and HD29 MTX mucus-producing cells in either 7:3 or 9:1 ratios. And this imaging showed distinct differences between the mucus production in the system. So these co-cultures enables the model to represent either small intestine with the physiologically lower mucus content or the large intestine with the physiologically higher mucus content. And since the main goal was to co-culture our gut-on-chip with patient-derived microbiota, we also established protocols for anaerobic viable microbiota isolation from the stool samples, and we confirmed it with the metagenome sequencing, and also we performed flow cytometry, air exposure tests, and live dead bacteria staining to confirm that we have isolated anaerobic bacteria. Before the co-culture of patient-derived microbiota with our gut-on-chip, we also evaluated barrier permeability in this model, and we saw barrier permeability decrease on day 5, and then it reached plateau phase, confirming functional cell layers suitable for microbiota exposure. Since these microbiota experiments are carried out under hypoxic conditions, we also evaluated the impact of hypoxia on this model, and we saw that barrier integrity increases, but there are no significant differences between microbiota and control groups, which implies that hypoxia itself is the main driver of the barrier permeability increase since the HIF-alpha-induced tight junction distractions. And lastly, we also looked at the cell viability, and we saw no significant differences before and after hypoxia. So then we moved on to the co-culture, and we added patient-derived microbiota into the top channel of the chip. And we cultured it for 48 hours and then performed metagenome sequencing. And here you can see top 20 most prevalent species into gut-on-chip replicates. And the main conclusion was that 70% of these bacteria were strictly anaerobic, while the rest of them were facultative anaerobic, and also 40% of these strains were mucus-dependent strains, which highlights that we have hypoxic conditions in this model and also mucus production. And what we saw is that if we look at the diversity, then in the chip replicates, the diversity is lower comparing to the reference sample. And that highlights the need for media optimization to reduce this selective pressure in this model and support higher microbial diversity. And after 48 hours, we also looked at the metabolomic analysis at the short-chain fatty acid metabolites, and we saw that microbiota, comparing to the control, is consuming nearly all of the glucose and pyruvate and converting it into lactic and succinic acid. We also saw changes in alpha-ketoglutaric acid levels, which implies microbially fermentative state and viable bacteria. We have also developed a wide range of different models and application cases. For example, currently we are working on the colon-on-chip models that will be further used for prebiotic testing, and we are also working on personalized models and models from primary cell cultures. Yeah, I want to say thank you to all of the collaborators and the team. And if you are interested more in this technology, you can contact me or we can meet after this session. Thanks for your attention.
Okay, thank you, everyone. So, as this is the first microbiome session in the PMNET Forum in the 4 years, it was quite busy. So I will ask all the speakers to join on the stage for at least short discussion. At least maybe there's some questions from the audience, so please. So are there any questions from the audience to the speakers in general or some of them in specific?
Maybe I can start with asking a question to both Mathieu and Marcus, because Marcus, you showed that there's quite some heterogeneity in the different techniques being used across studies, which makes it very difficult to provide recommendations. Now, also for these federated approaches, you also need consistency in pre-processing of the data using the same reference databases, read-based mapping or assembly-based mapping. How do you see how to move forward to reach this harmonization in the use of tools?
Actually, this is a hot topic, and there are multiple international initiatives that are trying to gather all the best protocols to optimize the microbiome analysis. There is the International Human Microbiome Standard that was created in 2012. There is the Human Microbiome Action now and multiple other initiatives. So far, the idea is there is not only one method that works, and it also depends on the ecosystem. The idea is to provide a list of methods that were tested and that are considered sufficiently good to answer some specific questions. For instance, exploring fungi, you need a specific method that is not applicable if you use classical metagenomic analysis. So it really depends on your question, on your ecosystem, on the age of the individuals that you analyze as well, because you have also higher difficulty in exploring low biomass samples as well, with a higher risk of cross-contamination during lab treatment, for instance. So these are considered by a major international consortium, and they tend to provide the best methods to help for these things.
Yeah, totally agree. So I follow the field for a while now, and it's constantly been people promote the idea of harmonized and standardized methods, but as Matthew pointed out, there are quite a few ways you can do the same thing and that are good practice. There are maybe even more ways you can do it doing the wrong way, so just being aware of which ones to have positive, negative controls to avoid and document confounders. We both talked about that in our talks. I remember going back to one of the first International Human Microbiome conferences in Paris and seeing the early initiatives, Human Microbiome Project and the MetaHit, and the comparison of those two, and they found two major clusters of healthy controls. One was Human Microbiome Project and one was MetaHit. So just as an example of big projects being very different. So yeah, record as much as possible, beware of the confounders, have plenty of controls, I suppose is my thoughts on it.
Okay, are there any questions from the audience? If not, then maybe just a couple minutes before everyone runs off to the lunch. I would like to ask, I think it's almost for everyone who presented today, we also yesterday had a discussion that it's great that we have biomarkers and we can say that microbiome can predict something, but of course the main aim would be to bring that to the clinic, to the patients, which is also like have been told in this conference many times. So what would you all see would be the next step, or what is missing, what would help you, or what's the vision, how to bring that the biomarker is really used by doctors in clinical practice and the patients understand that it's useful for them?
Well, I think, first of all, at least in our studies, we are mostly talking about associations. So I think we need clinical trials, which was also mentioned in a previous presentations. And the other thing is that, in a clinical practice, I think you need absolute values or some cutoffs. But we are mostly talking about some kind of relative values, so we should think about more straightforward evaluation of the microbiome components that we are seeing now.
I would say that we all know that microbiota is involved in many disease mechanisms, and it's important to study it also using cell cultures to look at these hosts' microbiota communication mechanisms. And organs on chips are alternative platform, and we have also some challenges with these microbiota experiments, but we are now moving forward more to personalized models where we can use also patient-derived cells together with patient-derived microbiota, which can help a lot of researchers and also patients in the future.
And something that we spoke about at the dinner, Ilze, is the cultural bridging between let's say microbiome scientists and clinicians, and I would throw in machine learning experts as well, that usually you have these studies of microbiome academic scientists who are great at what they're doing, and usually we add something, we have found biomarkers that could be used for diagnostic purposes, but we stop there. We don't know really what it takes to be a real diagnostic purpose. So I think, and likewise, you have clinicians who may not fully understand the microbiome science and how biomarkers are identified and what you can, and their limitations. So to have some sort of community where these groups can meet and understand each other would be a good starting point.
I can maybe follow up on that, because we had the same discussion also in the Netherlands last week about how to better communicate with each other, and I think what is missing now is we don't know how to develop these biomarkers, but clinicians indeed understand not enough about the microbiome, and I think what is important to incorporate also microbiome education in the medical curricula. We do that a lot in biomedical sciences, but not so much yet in the medical field. So I think to better understand each other and to understand the role of the microbiome, that's also one step to take.
Okay, then I thank all the speakers, and on that note that we still need to communicate better, and there's a possibility at the lunch, so there's clinicians, physicians and scientists, so let's do that. And thank you everyone for joining the first microbiome session in the PMNET Forum.